The Hong Kong Polytechnic University, PolyU, leverages its world-class academic and research excellence to develop innovative solutions for a more sustainable tomorrow. In July 2022, the PolyU Academy for Interdisciplinary Research PAIR was established as the university's central research platform, dedicated to providing world-leading interdisciplinary solutions to address significant societal challenges. PAIR is composed of multiple constituent research institutes and research centers. Each research unit focuses on a particular research area and is joined by researchers from various PolyU faculties, schools, and departments. PAIR research areas include advanced technologies and manufacturing, good health and well-being, and smart and sustainable cities. PAIR researchers develop new materials, systems, technologies, and techniques that support industrial modernization, upgrading, and transformation, look into the factors and mechanisms influencing human health and wellness, and develop evidence-based solutions, and generate smart technology-based solutions for making urban operations and services more efficient, connected, and eco-friendly. We particularly value impactful collaborative activities in research and scholarship. PAIR researchers pursue research collaboration at different levels. In addition to interdisciplinary collaborations within and between research units, PAIR and its units establish partnerships with industries, governmental bodies, universities, and other external stakeholders. With internal and external collaborations, we have successfully translated PAIR-led innovations into commercializable solutions and practical applications, and established joint laboratories and research entities with external organizations, driving scientific breakthroughs, innovation, and technology development, and socioeconomic prosperity. PAIR's research excellence and research impact have been widely recognized at local, regional, and international levels. Our research outcomes have been published in leading journals, including Nature and Science. Innovations led by PAIR researchers have won prizes at various competitions and events, including the International Exhibition of Inventions Geneva. Many PAIR researchers have been named among the world's top 2% most cited scientists in their own areas of specialty. Individual researchers have been selected to the National Academies of Science or Engineering and as fellows of professional associations, and have been bestowed with local, national, and international awards and honors. Harnessing the strengths of PolyU's research and world-class scholars, PAIR will continue our unwavering efforts in research and scholarship, knowledge transfer, and collaboration to develop impactful solutions that benefit Hong Kong, the nation, and the world.
Good morning, colleagues, ladies and gentlemen. Welcome to this seminar. My name is Qingyan Chen. I am the director of the PolyU Academy for Interdisciplinary Research, or PAIR. It's my great pleasure to introduce today's speaker, Professor LAO Lixing, President of Virginia University of Integrative Medicine in the United States. Professor LAO is a very close friend of PAIR. Has been serving as a PAIR fellow for many years. In addition to that, some of you may still remember Professor LAO gave another distinguished lecture 2 years ago that was very well received. So Professor LAO has a very distinguished career. I just wanted to mention a few of his achievements. For example, he worked very hard to develop this acupuncture in the United States, and that one now is accepted as a general medical treatment in the United States. And it's a very difficult one because for any Chinese medication, it's very difficult to get through the certification from the Federal Drug and Food Administration, or FDA. but Professor LAO's contribution really make a very difference. In addition to the acupuncture, Professor LAO also working very hard to promote like Chinese medicine such as Tuina, Tai Chi, and Qigong. So maybe during lecture he will try to do something for us. Okay. So at the Virginia University of Integrative Medicine, he teaches mentors, clinicians, and advanced in the field through evidence-based research. With more than 40 years of clinical experience, he has led numerous trials and published extensively on chronic pains, stress-related disorders, and internal diseases, make a lasting impact on evidence-based complementary medicine. So Professor LAO is a strong advocate for the whole person healthcare, emphasizing on the integration of rigorous scientific research with the holistic principles of traditional Chinese medicine. His outstanding contributions to the field has been widely recognized. In 2019, he received the Chief Executive Commendation for Community Service in Chinese Medicine from the government of the Hong Kong Special Administration Region. So without further ado, ladies and gentlemen, please join me in welcoming Professor LAO.
Thank you. Thank you very much, Professor Chen, for the introduction. I'm Dr. Lixing Lao. I'm very happy to be here. Thank the PolyU pair program invite me here. I'm honored to give a lecture here. A very fancy building. Thank you. Okay, this is the second time I give lectures. So as I mentioned earlier, that Professor earlier, I have been conducting acupuncture research for many, many years. And 2 years ago, I present my clinical trials on acupuncture. But today, as a holistic medicine, we not only work on acupuncture, we also work on the other modalities of Chinese medicine, which is herbal medicine, which is largely practiced in China for 1,000 years. So we make a big effort on the clinical trials in the United States. So let me start my slides. Okay. So today we discuss a couple of issues. One is the current usage of herbal supplements in the United States. Just give a brief how popular herbal medicine in the United States. But the herbals may be different from Chinese herbal medicine. That's a local medicinal herbs. And also I introduced 2 clinical trials. One is conducted at University of Maryland School of Medicine many years ago. And also more recently, I conducted a study with my colleagues, Dr. Wen Wei in School of Chinese Medicine, University of Hong Kong. So the 2 studies I want to introduce to you. And also after this, I can introduce the status of development of Chinese medicine clinical trials in the United States. And also the challenges, difficulties of these clinical trials. So we can discuss, open for discussion. I hope we can learn each other, okay? So let's talk about this from National Center for Health Statistics. They have a figure to show about 15 to 38 million US adults usage of herbal supplements. And the number largely varies because it defined what definition of herbal medicine and have different number. And the recent data also showed that 24% of the US adults used herbal medicine supplement or tea in 2021 to manage their health issues. And use the population roughly 1 in 5, okay, 20% of the US adults using the herbal products in the past year. Also, demographically very interesting, mainly elderly people, women use more than men. So this is what you can see. So let's look at the chart. See, in the left side, all different age, from 20 or over, 40, and 45 to 59 and 16 over. So you can see elderly people use more herb. And also compare middle, the man, the last panel, woman use more than a man. Okay, so women take care of health more seriously. And also very interestingly, there, I think the middle part, 20 to 39, not use much, but 20, age of 20 use more, then I think they have more time to consume herb. So the age of 20 to they're very busy their career. I don't have time, spend time on herb medicine, I guess. Okay, this is the chart also by the same center. They show the US used alternative medicine to treat health problem as 2021. So you can see herbal supplement usage is over 24%, okay, very large. If we look at the acupuncture in the down there, only 9% compared, even though largely used, still growing, but still herbal medicine more popular, okay. But of course, their herb again may not necessarily Chinese herbal medicine, including lots of herb. So then let me introduce our two centers, the Centers for Integrative Medicine or Center of Excellence for Research on Complementary Alternative Medicine. We were lucky founded by NIH from 5 years, from 2005 to 2010. So $6 million of US dollar cover 5 years program. So this aim of this Center for Grant is examine the safety, efficacy, and the mechanism of action of TCM treatment modalities, including acupuncture and herbal medicine for osteoarthritis, for arthritis condition. So here are the 6 parts, components of this center grant. They have a supporting course, so a botanical course led by, I think everybody knows David and Harry Fang, Dr. Harry Fang from University of Illinois. They take care of the botanical measurement. Then second with middle is Dr. Brian Berman, who is the PI of the grant, administrative core. And also we had data management and statistical core with a couple of people there, a couple of research. Then always have research project, 3 research projects. Number 1 today I'm going to discuss is phase 2 clinical trials led by Dr. Brian Berman. I was co-investigator. So 11 herbs of the follow Shaolin Dan, 11 herbs formula. Then the second part is preclinical trial using animal models to examine the same herbal products in animal models of arthritis, in animal arthritis. Then the third core led by me, acupuncture animal study on mechanisms of action of electroacupuncture. So then at the same time, we were lucky in the same 5 years period, we got another funding, center funding, international funding, International Center for Research in Complementary Medicine, collaborated with Chinese University of Hong Kong. So I don't want to mention it. Dr. Joseph Song was the co-PI with Dr. Brian Berman. So same time, we have 2 center grants. But today I'm going to focus on the first center grant. So the first center grant, we mainly examine a phase 2 study, examine the safety and dose effectiveness and double-blind, placebo-controlled clinical trials. So I have multi-site involved, University of Maryland, Baltimore, and also McLean Hospital, Harvard Medical School, University of Illinois. These 2 are mainly for the herbal quality control or quality assurance. And also we work with the Chinese University of Hong Kong, Prince of Wales Hospital to recruit patients. So why we choose this, our center have been long time treating the pain-related disorder. So osteoarthritis is one of the issues that bother elderly people, is a major cause of the mobility, physical limitations, and they're associated with the healthcare issues, leading cause of the disability of elderly people. And limited, most important, limited conventional analgesic treatment because they have concerned the possible side effects, the safety issue. That's why we're looking for the alternative treatment to see whether herbal medicine can, in addition to the Western medicine, can help this population. So the truth of the Horu Shaolin Dan is a classic herbal the Qing dynasty developed. Originally only 4 herbs, we modify into 11 herbs. So this is widely used for Chinese medicine as for treating for the B syndrome, we call B syndrome, also arthritis. And no previous study has been conducted to examine this formula. And the objective of our study is to determine whether this herbal formula improves the pain and function of the patient with arthritis of the knee. And while they continue to using the Western medicine, standard analgesics or NSAID, non-steroid anti-inflammatory drugs, and to identify the optimal dosage for this herbal formula and to see is it well tolerated by patient and safe for the patient. So then first we organize the research committee. So you can see the large team, and it's a major core, but more people involved. We have the experienced herbalists to make sure the herbal formula is suitable for the arthritis. And we have experts in the botanical preparation and analysis. As I mentioned earlier, they provide the source of the herb and sterilization and preparation of the herbal formula. Then we have this disease specialist, the rheumatologist, very familiar with the arthritis. And also clinical trialist, how to design clinical trial. Then we have data management expert to manage data, and biostatistician. Of course, we have the research coordinator and research nurse on different sites. Then the roadmap to reach to clinical definitive trial, phase 3 trial based on the FDA definition of the botanical research from bottom up. First, we have to identify herbal formula, which is suitable in the clinical usage. So the bottom you can see there. Then we have identified this formula, authentication, identification of this formula. Make sure that truly the formula what we want. And also we have used the herbal extraction for different methods, extract the herbal ingredient outside. Then we have quality control and quality assurance, make sure everything is stabilized, okay? And a long time, no significant degradation of this formula. And then we have to conduct preclinical studies with animal models mainly. And after animal models, then we have to apply for FDA, IND, investigation of new drugs application. And after approval, then we have to get approved from the IRB, which is the Institutional Review Board from the University of Maryland. Then we can launch a phase 1 pilot study. And if a success, so then we can move to phase 2, those escalations, phase 2 study. And if successful, then we reach the definitive trial, phase 3. Okay, this is the roadmap for all the drugs, and this is a particular for herbal medicine. So then we discussed the herbal formula. This is all 11 herbs. We use this formula. If we see the underlying herb is original formula. Original formula is Lu Xiang, Dang Gui, Dansheng, and Mo Yao. Mo Yao the other one. We removed Mo Yao because it have similar function with Lu Xiang. So we think it's overlap. Then we add other herbal formula can mainly promote blood circulation in the blood circulation and decrease the pain. Some of them. So first we identify the plants. This is the plants. We work with the China company Tongrentang to go to the field to identify the source. There's the plants we use. And then also we identify the parts of this particular plant. Which part of plant, because not every part of the plant have same function. We identify particular plant parts and the component of the plant. Then we go through the quality control, quality assurance. You probably, if we do research in urban medicine, very familiar with this also. TLC, HPLC, and LCMS analysis for further analyze this chemical composition. Then we get to the fingerprint. It's the fingerprint of the so we use this reference for the batch-to-batch comparison. Make sure consistent every batch, because this study runs about 3 years. So make sure each batch is similar, no variation. And also Harry Fong took picture from Changsha and GMP certificate facility to extract all the herb. So this is the eventual final products of the herbal the capsules, because in the United States, it's not easy to have the patient to drink the very bitter taste of the tea. The capsules easy for them to swallow. Then we, same time, the center also conducted the preclinical studies. Then we have preclinical studies. The two models we used. One model was the persistent inflammation model. Use this same herbal formula. So objective is to see whether this formula therapeutic effect in animal and safe, and determine dose effective risk. We have different dosage to see those relationship with the effectiveness. Explore mechanisms of action in neural pathway, how they through the neural pathway decrease the pain. So method we use this method, first way extraction, but we use different from usually in clinical usage. It was water, aqua only. They use acetone together, 70%. So we can extract more both polar, bipolar chemicals outside the herb. Then we have quality control, as I mentioned earlier, HPLC, MS. And also we use the formula published in the paper convert from the human dosage use to the animal use. Then eventually we come up with 2 dose, 2.3 per kilogram per day, and double it, 4.6 gram per kilogram per day. That's the dosage we use for the animal study. So this is also the major herb Luxiang, the first herb boswellic acid, PBC extraction. So trace the BC, because we believe BC is the major ingredient in this formula, more potent. Then also at the MS reflection, from this reflection to the MS analysis of the BC. And we trace of the HPLC, HL, the whole formula. Okay. Those are all fingerprint. And method we used for the animal study, first we introduced information on the animal for called CFA injection into the hind paw of the SD rats. Okay, group is 8 per group. And we measured hyperalgesia, the pain. Use the thermal stimulation. I'll show you the picture later. And we call paw withdrawal latency. The longer latency, the less pain. Okay? And measure information by measure the thickness of the paw. Okay? And the time we usually up to 7 days, from 1 day to 7 days, we measure every day almost. 0 day, 2 hours, 5 hours, 1 day, 3 days, 5 days, 7 days after the herbal ingestion. Then herbal preparation, we just use the sesame oil as a vehicle, as garbage to orally inject to the stomach. And we also use try to find the maximum of action for neural pathway using spinal cord force protein stain to determine the force expression in the spinal cord to see the pathway. And the animal protocol approved by IACUC, Institutional Animal Care Use Committee, Indian School of Medicine. So here the device we purchased from the University of San Diego to shoot the heat from bottom to the animal. So animal is restrained in the box. See here the heat, we push the button, the heat will shoot in the aim to the hind paw. If we regulate for 10 seconds, they withdraw from heat. But if you hyperalgesia, take only 3 or 2 to 3 seconds, they withdraw quickly, okay, indication of the hyperalgesia. Then the machine will document the seconds, and we measure the pore thickness to determine the swelling, level of swelling information. So results, very quickly go through results. The red line is the treatment group, the white line is the control group. We can see the treatment group significantly better pore withdrawal latency, the longer pore withdrawal latency compared to control group in the 24 hours before we measure before the injection of the herb and after, and each day, and 3 days, 5 days, 7 days, 5 days mainly, up to 5 days. Both improved. So then we measure the thickness. This low dose, 2.3 gram per kilogram per day. We use the pore withdrawal, pore edema. So the red line is the control of the treatment group, have less thickness. The white line is control group. You can see significant difference. So if we use a high dosage, 4.6 gram per kilogram, we can see also the treatment group better than control group. You can see the particular for this point, 24 hours, 3 days, and 5 days. And edema also, the treatment group better than control group when the edema, the decreased edema, the swelling. So if we look at the spinal cord, you can see the lamina 1, lamina 2 in the medial side is expression mainly C fibers through this area. So you can see lots of expression. If you look at the first one, CFA induced the pain, but only vehicles, no herbal medicine. Lots of expression in the medial side, lamina. But if we give the 0.5 gram per kilogram of the Luxiang, you can see decreased. Then if we get high dosage, it further decreased. The expression of the dot spots less. But if we see the herb only, no inflammation, no expression of the phosphoprotein. So this way determine the pathway through this lamina 1 or 2. So then we compare to the major herb Luxiang and whole formula in the spinal cord. You can see the control, the C-force, which is only inject about the CFA. And if you for CFA only, and also the inflammation, cause the inflammation. When control means they give the vehicle injection, okay, but no herbal medicine. But if you compare to 0.5 gram per kilogram, and no significant, but if high dosage, 0.9 gram per kilogram, significant, and 2.3 gram, which contain 0.45 gram, half dose of the Luxiang, show effectiveness. The formula is better than the individual herb, as tells you, even same dosage. Okay, then we see another model. This is the model we used for persistent information model. Now, another model for systematic whole bodies are called adjuvant arthritis model. This model involved not only limited the hind paw, but also all the joints of the animals involved. So this, we inject the CFA into the subcutaneously in the base of the tail to release the rat. Okay, and then this take about 8 to 10 days to develop the multi-joint arthritis. Yeah, we score to determine the severity of joint involvement. So results you can see that the red line, middle is red line, 2.3 gram per kilogram, and orange line, bottom is 4.6 gram per kilogram, compared to the yellow line, which is control, significantly improved. You can see, I think we started the 20 days because we wait until develop the arthritis, then we give the herbal medicine. Then you can see we started the herbal medicine and show difference with the score, the arthritis score. And also if we use the herbal formula for the computer control, pore swelling the pore also decreased. You see in the treatment group, both low dose and high dosage, but low dosage is less effective compared to the high dosage. So then we also measure the cytokines, and with pro-inflammatory cytokine, which is interleukin-1 beta and TNF alpha. Okay, so we can see from this figure, this treatment significantly suppress compared to control group, and no over-treatment or decreased significantly, either 0.9 gram of bosphoric acid or 2.3 gram of the herbal medicine and 4.6 with all significantly suppress the pro-inflammation cytokines. And this last one is normal animal, no cytokines actually. So conclusion is herbal medicine dose dependently reduce the arthritic score of the pore edema, and the maximum herbal medicine may involve to suppress local tissues pro-inflammatory cytokines such as TNF alpha and interleukin-1 beta. So then we also did a toxicity study. We have two toxicity studies. One's chronic, one's acute. The chronic has taken 6 weeks, low dosage, long time, and also acute for the larger maximum feasible dosage ingestion. But both chronic and acute show the safety, no difference between the vehicle control and the herbal medicine. Okay, both are safe. So then we move to our, based on the data from the animal study, from preclinical study, and then we move towards the clinical trials. The phase 1 study, we get the IND approval by FDA, and we give 6 weeks of the phase 1 study, 7 patients, and we found that they well tolerate the ingestion of the herbal capsules, and also no sign of the safety. And then we draw the blood test, chemistry, blood chemistry profile, all normal, and including liver function. Then we move to the dose escalation, placebo-controlled double-blind clinical trials, phase 2. So here is the trial led by Professor Brian Berman, and also I was the co-I. So this is the ratio of 1 to 2, 1 to 2, which is treatment to 2 and control 1. Okay, but actually 3 arms, 60 each arm, was because with treatment group we have 2, low dose and middle dose, 2 dosage, or 3 dosage actually. So this is the FDA approved ingredient for placebo. We have to make a placebo and work with the School of Pharmacy in the University of Maryland. And we work with our colleagues. They come up with the list approved by FDA as safe. And some of them to provide the color, a similar color to, and some of them provide taste, bitter taste. So make 2 groups very similar. So here you can see both formula very similar. If the patient don't have direct comparison, they cannot tell the difference. And I guess you can guess which one is real herb, which is placebo. Can you guess? It's easier to guess. Anybody? Left is real herbs. No, it's the opposite. Because the right side is rough. The herb is rough. If FDA chemical synthetic is very fine. So that's the difference. For herb is much rough. Okay. Thank you. Okay, the study design is 180, okay, total, and 120 herbal medicine and 60 placebo randomized. And we're actually, because we cannot get enough patients, so we reached to Hong Kong, Chinese EU of Hong Kong, to ask their help. And Dr. Lee in Hong Kong, Prince Well Hospital, they have lots of patients. So we conduct a study. But here the results, maybe you feel disappointed. See, if the intro comparison from baseline 8-week, the change, the p-value is more than 0.01 is significant. So no matter herbal medicine or placebo medicine, after treatment, all get improved in all the functions, warmic function, pain function, all different function, pain, all significant, p smaller than 0.001. Unfortunately, between group comparison, no significance at all, 0.47, 0.5. See, last column is between group comparison. This column is compared to baseline. So that means as soon as you enter the group, you're all getting improved, getting better. So this the finding we have. But also safe, of course, no any side effects were observed. So this is also the adverse effect. We find that no superior, the side effect superior to the placebo, no difference compared to treatment group and placebo. So people published the arthritis and cartilage. Okay. So lesson learned, I want to share that you can see animal studies show so effective, right? So good. Between group, very significant. But that means the animal gaps. So we cannot rely on the data on animal. The human is different from animal. The human have more variation, your mood, your emotion, other factors, so affect the human. Okay, and disease may not be appropriate because osteoarthritis is not as inflammatory as rheumatoid arthritis. Later on we discussed as the knee wear out, the joint wear out. The herbal formula may not be potent enough. Okay, so cannot be potent enough. But the herbal basically is not so potent as drug. We already know. Okay, the very natural products. And the fixed herbal formula without the modification for the individual treatment may be different. This is also the limitation. We did not use any differentiation based on individual patient. We all same formula. This might be another issue. So maybe some patients get benefit, some people not get benefit. When average, no benefit. But if you look at individual, you can see maybe some benefit. So in the future, maybe individualized formula based on TCM differentiation may be needed. Okay. So this is the way we discussed the lesson we learned. But we're not alone. If we look at this slides, New England Journal of Medicine published negative study. You can see all herbal related to the individual herb. Okay, this let me see. This is the Soporto for the benign prostate hyperplasia. Okay, enlargement of the prostate, and this is for the glucosamine for arthritis, for the arthritis. They all show negative study, no significant improvement. So we're not alone. So then, as if we continue to see JAMA series, in the fourth study they published with St. John's for the major depression have been used. Still, the market is still sells the same product. And vitamin E for cancer-related trial, and the ginkgo biloba for the dementia, and as well as the mixtoso. So also show the negative study, no side effect, because there are large clinical trials at that time. So let me introduce, this was happened in the United States. Then since I work here in Hong Kong, University of Hong Kong for 6 years, I tried to make clinical trials here and use slightly different approach. Instead of use isolation, extraction, we just generally regularly practice, like decoction, just to take the tea, the soup for the patient. So this is led by Dr. Manwei, Dr. Man. So this is for the disease, for the tumor shrink distortion, okay? A Chinese formula to the symptomatic uterus fibroids, okay? Dr. Man is a very well-known the gynecologist, Chinese medicine gynecologist. So we use this model because Dr. May has experience using this formula for lots of patients. And we know this came from women's issue, which the benign tumor in a woman account for 30 to 40% of women population. So major symptom, irregular bleeding, vaginal bleeding, and frequent urination, and urinary retention, abdominal pain, symptoms. So we use this formula, individual formula, then you can see the composition formula. So because in Hong Kong that time we were told we're not allowed to use the placebo. We're not, if placebo had to go to very risk procedure. So we decided to use low dose compared to normal dose. So low dose is 1/3 of the normal dose. You can see about 1/3. So instead of 30 gram, we use the 10 gram. So everything is reduced to low dose. So we want to see whether the normal dose has more effect than low dose. The hypothesis of the study objective is that this formula can reduce the size of fibroids and improve the symptoms, improve their symptoms. The high dosage will be better than the low dosage. That's what the hypothesis. And the objective is to evaluate efficacy and safety of this formula in disease and test the high dose, with high dose better than low dose for treated. So here is randomized clinical trials. We use the high dosage, totally 217 gram, low dose totally 69 gram, and for 16 weeks, a long time treatment. Control the high dosage versus low dose, no placebo control. So this is the slightly difference. Then you can see the timeline for long time follow-up, and the primary outcome was measure the severity of the uterus fibrosis-related symptoms, okay, and health-related quality of life. And secondly, we measure the size of the fibroids, is the MRI. Okay, so randomized, blinded, and this is all very standardized. Computer-generated block coding, double-blind. Patient, they don't know which one. They both the herbals same taste, right? Only the dose is different, so patient cannot tell the difference. So we get the IRB approval and trial registered and patient recruitment. And also we established a data safety monitor board. I think Hong Kong that time didn't have this board. I don't know if now they have the, in the United States, every clinical trial you must require independent DSMB, which is not part of your university, somebody from outside. You must meet regularly to examine your study, okay? And then we have a weekly meeting to follow up. So participants, 78 subjects, and we have a long list of the inclusive criteria and exclusive criteria. I'm not going to go through, but you see the serious disease we excluded. Okay. And herbal quality control, we have the Dr. Zhang, Yanbo Zhang, also help us to, and also we use the herbal Hong Kong Authority Certificate Herbal Chinese Herbal Supplies via the School of Chinese Medicine School, the herbal dispensary. And by active component detection of HPLC also used. And everything else we monitored from time to time, measure the decoction to be consistent, make sure consistent. We have a machine to make the boiling machine come up with the formula, the tea. So here you can see the HPLC for control, and the retention time, so the time to see any degeneration, degradation. So then also primary outcome, 37 items instrument usually to relate to the severity and quality of life, okay? So I'm not going to go further. Okay, now use MRI scan to detect the size of the tumor, fibroids in the abdomen, okay? So this is the secondary outcome measurement. And also determine the the hormone level and blood sample, blood chemical profile, and at the baseline and end of study to see any difference. Okay. And particularly estrogen, FSH was measured. So then DSMB will meet currently, so and knowledge a couple well-known doctor in this Hong Kong, Dr. Bian Zhaoxiang also in the DSMB, Vivian Wong also in this meeting. So then the results may not very promising, but you see the low dosage and high dosage compared to baseline, they all significantly improved. This the before and after treatment. So the severity of symptom decreased after treatment, and the high dosage also decreased, and the low dosage also before after. Okay, but interestingly, the low dosage seems better than high dosage. This maybe can save us, to give us some lesson that we don't use that much of herbal medicine can still achieve the same results. So you can see if we compare between two groups in terms of the primary outcome, no difference. Okay? And it's slightly different, but no statistical difference. No statistic. And yes, the upper line is low dosage, lower line is high dosage. So the only significant difference is the TCM symptoms. TCM symptoms, you have lots of questions for TCM, sleep, everything, also quality of life, also the pulse, diagnosis. This is blinded, so the practitioner measure the pulse and time, they don't know which group assignment. So this shows the difference between two groups. Everything else is the same, no difference. And also secondary outcome measurement and size of the tumor, also fibrosis, no difference between two groups, okay? And also adverse effect, no difference, and no side effect can be detected. So it's safe, but no effective either. So the study published in Journal of Anatomy Pharmacology. The Man Wei is the first author. So lesson learned from this, because we have no placebo control. So we really is not really compared to the Endowed treatment. So we don't know if this low dosage also effective. We don't know the dosage. And low dosage have similar or even better effect than the high dosage, because it gives us a lesson that we may not need to use high dosage for the patient care, so save lots of resource. And we have the batch-to-batch consistency. We measure each time, but it's difficult. Compared to previous study, it's chemical extract, so very stable because it's batch-to-batch. You have the long term, you have to buy new batch from the market. So each time maybe slightly different. Because again, this is the fixed formula without modification, without TCM differentiation. It might be difficult. Okay, then after discuss two studies, hope not too pessimistic. We discuss current status of clinical trials in the United States mainly, but even including worldwide, because people register in the United States. I've searched the PubMed with the keywords of the Chinese herbal medicine and the randomized clinical trial. From the 1996 to 2005, only 213 papers published. And later on, getting more, 2006 to 2015, more. So last 10 years, have 1,000 publications. So you can see the currency trend is more and more people like to do studies. So this is compared to previous 26 years to recent 10 years. Previous 10 years, we have botanical study, 747, but herbal medicine, 307, about less than half. But if we look at the past 10 years, 500 botanical study conducted and registered from the NIH clinical trial .gov, and among them, 207 study registered, but only one with results. The other one doesn't have results. We don't know, they're not reported yet. So JAMA, but more promising, recent JAMA large clinical trial be published, very positive, is which 2 years ago, 3 years ago, 2023 in Burma, they used the herbal called Tongxin in for patients with acute myocardial infarction. So this study shows significant compared between two groups. We can see primary outcome 0.0, points more than 0.06. And almost all the significant, only the one enlargement of the coronary, this third one not too effective, but very promising. But the other hand is mixed results. This study also used Angong Niu Huang, very famous Angong Niu Huang, 2025. It's very recently published paper. They show no difference. See, p very bigger than 0.05. So it's lots of challenge. Our mixed results is very confusing. We don't know what's going on. So that's what I want to share with everybody. Can get discussion more to see how to address these issues. And now look at the FDA approved botanical drugs. Only 4 so far, not over formula. Only these ones from something from green tea, for external genital and the warts. Okay, and also the other one is from the tree and some things of pineapple plants, a stem of pineapple plants from tea bark or tree bark. 4, okay, this FDA approved, but none of them associated with Chinese medicine. So finally, I acknowledge both Center for Integrative Medicine and your environment. Thank you, NIH give us funding for the negative study. Thank you. And we have also acknowledged the study conducted in Hong Kong, provide MHRF, okay, Hong Kong government, and also the person involved the study. Also, I would appreciate their support. Vivian, Wong, Professor Ben Zhaoxiang, many other people. Thank you very much. And time for Q&A. Thank you. Maybe I should sit there. Panel discussion.
Okay, Professor Lau, we have some question for you now. Okay, well, by the way, anybody has a burning question first? Okay, Vivian, we'll let her ask you.
Yeah, thank you, Professor Lau. So it's great to hear the negative results. Give us some confidence that as long as we conduct the clinical trial strict and scientifically, negative results is okay too. I'm just wondering that for your second trial, because it's a bit confusing to me. The N number is like 39, if I notice it right. It's not really a...
78.
No, the second trial, the
In
one
the-
with low dose and high dose.
Oh, yes, the first study, right? First trial in United States.
No, the one in Hong Kong.
Kong?
Yeah. I saw that in the table it says the N number is 39, and the one in United States is like 60 or 40 something.
160, yeah, 160.
Yeah. So the N number is not like, because we usually need to calculate the power, and even especially this is not a placebo-controlled clinical trial. It's a low dose and high dose. The N number is not that much. So I'm just wondering that at the very beginning, you can already see the limitation. Even the results come out, you cannot really conclude
No,
this herbal is effective or not.
Both studies we estimated the power. Use the
Oh,
power
okay.
Because if no estimated power, you can get funding. You have
I
to
see.
demonstrate the power based on previous studies. So
Okay, so even 39 is good enough. I see.
39, I think 39 only partially. You measure probably fibroids.
Okay.
39. Total 78.
Oh, okay, I see. So for the low dose versus high dose, that clinical trial, I'm just wondering that even before the clinical trial, you can already foresee the limitation of this study, right? Because even the results come out, you cannot really conclude the herbal is effective or not because you don't have a placebo group. So I'm just wondering that at that time point, why or how, what kind of factors made you make the decision we will continue with this trial, even this is not a placebo-controlled trial
It's very simple because Hong Kong not allowed placebo-controlled trial.
at that
That's
time?
simple. That's right. I think it's now still the issue.
No,
Be-
not now. I
You can
think
use the placebo.
yeah, we are
Yes.
doing a clinical trial now, and it's placebo-controlled.
Okay, that's good.
Okay.
At that time, they not said not allowed.
I see, I see. And the other thing is that for the clinical trial you conducted in Hong Kong, you didn't really do any animal study, directly jump into the clinical one, right?
We didn't do animal study. That's right. Because
Okay.
this has been used in the clinical already for
Oh.
patients already.
Oh,
The
okay.
doctor man using the daily basis for patients. So we don't need to go back to the animal study.
Okay, so this is herbal already used in the
Yeah,
clinical trial.
yeah.
I
The
got
reason we get approved because we in application we say daily use. Even the way in the trial, we still patient coming to clinic, we prescribe herbal for them. They're not pre-made, not pre-made. They
Okay.
prescribe for the herb, and patient will go to the herbal dispensary, they get same herb. And this also we make the double blind. Some patients get low dosage, sometimes get up to high dosage. But
Okay.
the doctor prescribed the herb, they not know this patient will get which dosage.
Okay, I see. So that gives you some reference that how to define low dosage and high dosage as well, right?
Yeah,
Because it's
because
already in the
he
clinic.
just give if this patient agreed to be participate in study, so he just prescribed a formula. He doesn't even, mainly he measured the pulse, time diagnosis, questions, document. The formula is the same, right? Then you give to patient. Then whoever get this code, we disclose the envelope, the number of the randomization code. If this randomized happen to be low dose, the herbal dispensary, they give the low dosage.
Okay, got it. Thank you. Thank you.
This is how we did double blind.
Since Vivian already started, anybody want to follow her to have any questions? I just want to ask or comment on this low dose issues because we have been doing animal studies all along with herbal medicines, and it's always not dose dependent. And we find many times low dose is better than
Oh, yeah.
high dose.
Really?
What are the reasons? Then of course you can always go into more mechanistic thing. And also now we have this gut microbiome issues about how you change the system and all that. And I will ask those questions later. So just follow up because I think for in effectiveness, one of the concern or issues is whether all the patients have the similar body type. Because for both of the clinical study, you're using symptoms or a disease that a patient diagnosed from a Western point of view. But that doesn't mean that each of the person who is suffering from the same diseases have similar body types. Can you comment on whether those two formulas could be prescribed as a standardized preparation because everybody's are different?
That's a very good point because we know in the Western diagnosis criteria determine the diagnosis, but in TCM point of view, there are many different types. Could be insufficiency, could be young deficiency, could be a different stage of disease also show. In the Western medicine, same diagnosis because the parameter is same. But that's why in our lesson suggestion is that you should, based on individual, should tailor the formula, should be modified the formula to fit the individual condition because it's all different.
Yeah, that would be like more personalized prescription, but could we also analyze based on like subgroup analysis? I mean, this is very common in nutrition interventions. you probably cannot control whether a person smoke or working in whatever environment, but if you can do a large enough study, then you can kind of subgroup to analyze the different types of body type,
Yes.
could probably help to explain.
Yes.
Yeah.
But unfortunately, this must be, like you said, it must be large enough to if only a few patients,
You can,
even your sub-analysis is not meaningful, not significant. So this is the challenge for us. But
Yes.
your sample size calculation is not based on sub-analysis, it's based on the entire group. So you won't be exaggerate the sample size. You have to use the appropriate sample size.
So if we design a new study with the input from the Chinese medicine kind of categorizing people, might be able to answer that question.
Yeah, then you have to enlarge the sample size based on
Right.
sub-analysis.
Okay. Now I have some question here that is prepared ahead of time. What do you think about the anti-Chinese herbal thoughts on the internet? I mean, this is a very general thing that some people don't trust TCM at all. Do you, what is your comment on that?
First of all, I've never watched this kind of, I don't know what they're talking about. I think a couple of things, issues. One is issue that so far no any very clear evidence, I think, so make them have the weapon to against Chinese medicine, because you see, you don't have evidence, right? You can see that. So we need to work hard to produce evidence. That's the one. And the other one is I heard that some of the TCM practitioners exaggerate the effectiveness of treatment, makes very large. So this also not very good. We have to base on the facts, whatever happens, and we can work together.
In contrast, because I think last week we have a symposium. In fact, it's interesting to hear from the Harvard scientists. They say actually Chinese are the one that is against Chinese medicine, but the Western people now are very into this body work, like Tai Chi and all that, and it's become very popular.
Yeah.
Oh, what is your comment?
Oh,
with
yes,
that?
I heard a lot about that. When we had conference, that early times we discussed about acupuncture even, we not talk about medicine. We heard from the Western medical doctor in the conference, they told us the story when they have a group of doctors together and they find that the Chinese doctor in the same group fight each other. And the Western doctor, Americans should persuade them not fight. Please calm down. That's the issue.
So what should we do in terms of evidence to bridge the gap? There must be some reason why these people are fighting, right? What do you think about the evidence based on herbal medicines versus those from the Western medicines? Are the two really against each other?
No, not really. Just for me, is that if you look at the human disease, Chinese medicine, Western medicine from different angle, look at same disease, everything's same. So we need the integrative, then you TCM practitioners should understand the disease, the Western, but they don't use the Western approach. Like you fixed formula, this is the Western medicine approach. You should be used to differentiation, I think. TCM, many years we always use differentiation, individualized, but now towards the Western styles, Westernized, then it would be difficult. This, I think, is a limitation.
So we just mentioned about the challenges, right? Because TCM has a very different theory behind whether it's a body work, whether this is herbal or even food, in terms of how we tackle a systemic problem. So would there be any chance that we can have a consensus of how to design better study? Because if we use the traditional Western way of doing research, we probably would never be getting to a certain destination, right?
I have a couple of lectures and also papers talk about this issue. If we look at back, Chinese medicine practice, they never use the one individual herb, right? I never use the fixed formula for other people. If you really want to study traditional medicine, you have to follow the many years of traditional medicine, which is individualized differentiation, right? So I often give examples, like Western medicine, if like a door locked, a door, you want to open the door, the Western medicine is very potent method, very strong, but sometimes cause the side effect also very strong, right? They can the hammer will knock down the door, lock to open the door. But Chinese medicine sounds like a key. You make a key, you have to fist exactly into this lock and open door more gently. But it's not very potent. The botanic herb, lots of herb we eat daily, right? Lots of ginger, garlic we eat every day. It's not for treatment. But if a good combination based on individual condition, you have good, remarkable effect. So how to resolve the issue with a couple of difficulties. One is if we want to prove the concept, you want to say Chinese medicine works. Okay, you have to individualize. I have the design, propose the design. The patient can be same disease, could be same disease, but the practitioner should be prescribed different formula for different patients. But still can be blinded. Okay, some people randomized control, you can give a placebo. Okay, but the herbalist give the same formula for this patient. But then in the back, if the herbal dispensary find this code, the patient should be randomized into clinical trial, they won't give the real herb. So it how can differentiate. But each patient have different formula. However, this kind of approach, you may able to prove the concept that Chinese medicine work, but you cannot commercially, you cannot make a profit because everything they're saying, no herbal company interested in this kind of study. Nobody will get money because you cannot produce any fixed formula on the market. This is the issue we now face.
Since you mentioned about this approach, I'm thinking recently they call this real life, real world data. What do you think about using real world data versus doing intervention trial?
Yeah, this is now people talk about the real data. Means a lot of people using whatever practice use. So collect large data to analyze this one. But of course, you need a large sample. Many people join this study, and also lots of noise. You want to create noise as well. So there's also some challenges.
Those are more statistical
But
studies.
for people against the Chinese medicine, they always have all criticize. It's not very rigorous study, right? It's not very rigorously designed study.
I have one more question from our registrants about whether the herbal medicines can be confined to a particular region. I think the meaning is more environmental factors, like whether all the herbs or plants are available in a particular part of the world that might affect the effectiveness. And the other thing is whether the same medicine work in patients staying in another country, maybe because they're adapting to a different environment. So what do you think about the environmental influences of the efficacy of herbal medicines?
Okay, thank you for the good question. This is in the world, many places have different herb, right? Africa, America, they have local different herbs. Okay, but as I show the slides, lots of native study, they're all local herb, not Chinese herb, right? Okay, some water drones, they're also selective too. Okay, so even though they largely use, they believe they use many years, 100 years from the parent generation to generation, but in the clinical trial, it will be difficult. Maybe they also be individualized. But other effect for the promotion of the globalization of Chinese medicine into the Western world, the question I think mentioned, maybe because of cultural difference, they may not trust Chinese medicine. But I think the other way, some people even believe more because it's foreign, it's more fascinating. Okay, something I never heard, maybe very good. It could be either way. It's not a big issue. But I want to mention a little bit advertise for my Sunday's conference. You're going to join us for the wildlife protection in Chinese medicine. This is a very important key in Western medicine. People hesitate to take Chinese medicine because they think they are taking wildlife, endangered species, to kill wildlife. Because the pattern herb, they cannot tell what's inside. They're suspicious. So that's why we need to have this conference to tell people it's not the case.
I see. Okay. I have one more question from online regarding the properties of the Chinese medicine, which is multi-formula, multi-compositions, and also multi-target. Would this property affect how we calculate the sample size and also what kind of marker for efficacy detections? Would that, how do we design a study based on this kind of properties of Chinese herbal?
A good question. For any clinical trials, there are always lots of outcome measurement, right? Quality of life, pain, stiffness for the arthritis, for example. Usually we design a clinical trials, we are looking at the sample size. The sample is based on the primary outcome. You cannot base on every parameters, otherwise your sample size is endless, too big, right? So what you want to measure most means what you think this formula works better for this condition. For example, you think that this formula best for pain. You should use the pain as a primary outcome. The sample size should be calculated based on the pain. If you think as for the function, stiffness or whatever, then you based on the stiffness. This is how, no matter how many multiple choice targets, you only face the one or two main target, which you think most effective. That's how you should do.
So we should choose the most important target first.
Yes, right. Yeah.
That demonstrate the effects of
Yeah.
the herbal.
The so-called important target, 2 meanings. One is important for the herb, most effective. You make sure this herb can treat this condition. Number 2, for disease, Western medicine already recognize this is an important issue. For example, the people have we just give examples. People have a sleep disorder, like insomnia, right? You give herb, then sleep is not getting better, but their bowel movement getting better, appetite getting better. So Western community won't accept. So you have to, if you treat herb insomnia, you should emphasize the primary outcome should be sleep.
Okay. Now I want to see if there's any question from the floor. Yeah, oh, Chi-Wei.
Yeah, Professor LAO, thank you so much for your talk. Actually, I'm very new to this area, but we are very interested in learning more about the potential of TCM in treating eye disease. But for many eye disease, actually they are chronic in nature, and therefore the current management actually require a long-term and even like a lifelong therapy. So in the context of a clinical trial involving TCM, so I wonder whether it is ethically or practically feasible to administer, for example, a standardized TCM formulations over a prolonged period, or as you mentioned, maybe we should adopt like a more personalized approach. For example, you may have to adjust the dosage or even the compositions when we come to the design the experimental protocol, because it lasts for a long time before we can see the effect for many eye disease. Thank you.
Yes, I think that's a good question. This is similar to any other clinical trials from herbal medicine. The first step is we have to identify herbs. Based on what? Based on the daily base practice. Work with the herbalist who are familiar with the eye disorder, for example. He has experience. He used the herb for certain disorder and can see the results. So you based on this, then you can continue to choose which the disease, which form is the most appropriate to use. But in the beginning, you want to choose a short term. You don't want to choose long term. You cannot never finish or never recruit patients. You choose something easy to demonstrate efficacy first. Then from that point, you go to difficult step. That's my suggestion. Whether you change the same issue with other studies, whether you variation or individualize or not, the challenge face every day. Whoever you want to conduct an open study, you all have same challenge. Thank you.
Gentlemen, yeah.
Thank you, Professor. My question is, what are the major differences between conducting a clinical trial of a Western pharmaceutical and a TCM?
The clinical trials is actually basically the same when you reach the clinical trial stage, like a phase 1, phase 2, phase 3, no difference. The Western drugs, same phase. The only difference is the preparation stage. We have to take the herbal from field. You make sure the In the Western drugs, you have the synthetically, you can chemicals very easy to control. The botanical herb, you have to go to field and soil and heavy metal contamination. All these you have to make sure control. It's more difficult. And also different region, north part of southern part, they have different land, different soil, maybe produce different quality of the herb mass. This is the difficult part. But as soon as have this part go through, quality control, quality assurance, conduct, and give to human the same, phase 1, phase 2, phase 3, no difference.
So in that case, then we have to conduct a TCM basing on the source of the ingredient. So from the north, we conduct one clinical trial, and then the ingredient from the south, we conduct another clinical trial.
No, I don't think
mean that?
necessary. You can use all different regions as long as you show the efficacy. You have to north, west, east, you have together. If you show efficacy compared to control group, that's it. That's you demonstrate effectiveness. Then you want to compare north better or south better, then a different study called sub-analysis. Use the herb only north or only south. But this is even now the modern technology, you don't even to compare to different source. They can analyze chemical concentration. Some growing in the north have lower concentration on the botanical herb, but some in the south have more rich materials. That's all you can find already.
Thank you.
Okay.
Another question.
Yeah.
How to ensure the consistency in the efficacy of the TCM, taking into account the batch-to-batch variance of the origin of the ingredients?
That's good. Okay, so what we do is two different methods. The first method is if the study not too large, if like 100 people, 200 people, not too long, you can use one batch only. The same batch, large amount, you can storage. Then we have to do, before the study, we have to measure the ingredient to see if the degradation decline or not, if they stabilized. Okay, shelf life, then shelf life, if stabilized for 3 years, they still same, then you can use one batch consistency. But if there's something very quickly degraded, declined, you have the expiration date, right? Then you have to make a new batch. Okay, then what we use the fingerprint to match. You make sure new batch must very similar to the fingerprint HPLC in the first batch. That's how serve the reference batch-to-batch consistency. That's how we do it.
Thank you, Professor.
Vivian.
A follow-up question. So when you are comparing the batch-to-batch difference, you have the fingerprint data, you have each chemical, the peak value, right? So do you need to guarantee that over 3 years, the fingerprint did not change? Or do you need to identify which are the key components in this herbal and make sure those key components are not changed over 3 years?
Yes, exactly.
So
you
it's a
cannot-
fingerprint or the key
No,
component?
the fingerprint have so many
Finger-
fingers, right? So
Yeah.
fingers, right? You cannot measure every fingers. You have the key. If you think these major, like a phosphoric acid, like the Lucia is the major one, you play major role. This one cannot change too much. So other one is maybe modify, start, modify. But still, I think there's still percentage of 80% or 90%. You allow 10% of variation.
But
But
then
you
how,
cannot-
how do you define or identify which are the key components?
Oh, this before you start, you have to identify. You can, because this are already very well documented. Harry Funk can tell you better story. So it's documented already which part have inflammatory, anti-inflammatory effect, which is the anti-cancer effect. They already know.
So those are single compound.
Compound, yes.
Okay, I see. Thank you.
Inside the entire formula.
Okay,
Yes.
Okay, Daniel.
So Professor Lau, thanks for the lecture. So in the clinical study you conducted in US, the placebo effect and the TCM formula both works. So the placebo effect is strong and masks the effect of TCM in many clinical study related to TCM. So I just wonder, is there any way to remove the placebo effect? I think placebo effect is quite common in clinical trials. So for example, if you just tell all the participants they are receiving the placebos only, and so that when they receive the actual herbs, the effect may show up in the effect. So I just wonder, is there any study examining how to remove placebo effect, and whether that has been, do you think is effective using similar strategy in clinical trial of TCM?
Yes, that's a very good question. Actually, I have a couple workshops and couple large conferences only focused on placebo only, just discuss placebo, everything about placebo. So I can tell many stories about this. But in the end, the people believe that the best trial is placebo-controlled trial. You control placebo. So if you're better than placebo, means your products not only placebo, you have placebo effect inside. You must be better than placebo. See, placebo both the same, right? Both group. And if you're better, then means you have more than placebo effect. But if we tell them the other story mentioned about, tell them all placebo, we'll have another issue called nocebo effect. Have you heard nocebo effect? Nocebo effect means make worse, make condition good. If patient know they not get a real treatment, they decline, their symptoms decline, getting worse. So you don't want to happen this happen too. Okay. And not only placebo effect, also in the trial, they are called regression meaning. Regression meaning anybody enter the clinical trial, even no treatment, if they're waitlist, I'll give you treatment be 8 weeks later, now they wait. They can improve. The placebo regression to mean, so if statistically can show that. So lots of other factors. That's why so far still randomized clinical trial placebo control is still gold standard. No better method.
Now, for the interest of time, if anybody have a burning last question? Now, if not, then please join me to thank Professor Lau.
Thank you very much for your attention. Thank you.
Okay, well, thank you again, Professor Lau, for your very outstanding seminar. So to express our appreciation to you, we make this special plaque with your name and the title of the seminar on this way. Okay. So I also wanted to take this opportunity to thank all of you for coming here to this seminar today. And also wanted to express those who ask questions to make this seminar even more meaningful. Thank you a lot, and this seminar is adjourned.